Hello students, welcome to EPG Pathsala. My name is Anant Narayan Bhatt and I work as a scientist in ADRDO Lab, Institute of Nuclear Medicine and Allied Sciences, which is situated in Delhi. So today we are going to talk about protein stability and how we can calculate the protein stability, how we can understand about the protein stability, how stable the protein of choice is, and then how we can engineer it. So this module is the part of molecular enzymology and protein engineering. So when we talk about protein stability and enzymes, what is basically in the focus? The focus is on like how we can engineer them to suit the industrial or commercial requirement. So the enzymes are evolved to suit their natural habitat. They are not evolved to suit the industrial requirement. So they are evolved to suit the natural habitat inside the cell or system. So however, during the application of protein enzymes for medical or industrial purposes, the molecule have to face many adverse environments because they are being used not inside the cell, they are being used outside the cell and in a lot of harsh environments. So enzymes isolated from nature, they do not fulfill these specific demands. So they have to meet their industrial or medical application. So to meet their industrial or medical application, in most of the cases, stability of the protein in this new environment is a major concern. So therefore, the estimation of the stability of the protein using various denaturation techniques and their chemical modification of amino acids to enhance their stability and enzyme activity, etc., whatever is necessary, I mean whatever is the target, is the basic motive of what we are going to talk today. So in this module, we will study how to estimate the stability of the proteins using denaturation curves and how chemical modification can help in engineering the physical and biological properties of the proteins. So so we will be one by one we will be studying about the protein stability, and then what is the denaturation curve, how we can generate protein denaturation curve, and then how we can analyze these denaturation curves, and after analysis whatever the modification, whatever the chemical modifications we can do to make this proteins more stable or more active. So when we are talking about protein stability normally refers about the thermodynamic stability of the protein. So so these are called physical stability of the protein. Thermodynamic stability is basically called as physical stability of the protein. However, the chemical stability of the protein basically involves the cleavage of the peptide bond, not the overall stability or degradation of the protein. So it is basically involves the cleavage of the peptide bond, like by the oxidation or disulfide interchange at neutral pH, somehow it occurs, and then that is basically called the chemical stability of the protein. However, the biological stability of the protein is basically increasing the shelf life of the protein inside the cell. For example, like if the acetylation of the protein occurs at lysine residue, then the lysine residues cannot get ubiquitinated. So if the lysine residues are not being ubiquitinated, they cannot be taken for degradation through proteasomal degradation pathway. So this is called increasing the biological stability of the protein, or preventing it, or by modifying preventing it from the degradation of proteolytic enzymes. So we will be focusing mainly on these aspects in this module. So protein folding is important for many reasons. Studying protein folding provides an understanding of the basic thermodynamic process of protein folding, as well as increased protein stability is very useful for many things, like when we are using enzymes for industrial purposes, like for the production of some chemicals, or in biotechnology, or in any protein drugs like insulin, etc. So the stability of the protein matters a lot because it's a pharmaceutical compound. The stability is increased, and the half-life of the drug will also be increased. So it has industrial or pharmaceutical advantages if the protein stability is high. And if we study and we are able to with whatever protein engineering modalities, if we are able to increase the stability of the protein. So two major innovations has or two major techniques which were developed in recent past or in few decades in past, which impacted a lot the study of protein stability. The first one is differential scanning calorimetry, which gives the idea about the protein stability, how much stable the protein is against the thermal denaturation, and the site-directed mutagenesis, or a lot of computational techniques which can predict that if this amino acid is mutated with some other amino acid, then the stability of the protein will increase or it can be decreased. So based on application, if you apply both these techniques, then the protein stability can be altered, and that sometimes suits the industrial requirement. So when we talk about protein stability, most of the time we are talking about the thermodynamic stability of the protein, because from the industrial purpose, because when we are taking out the protein from the system, from the cell, then its stability is considered at room temperature or at whatever temperature its reaction is required. So we are talking mainly about the thermodynamic stability of the protein, which is the physical stability of the protein. So in thermodynamic terms, the stability of the protein is the difference between the unfolded state and the folded states. So the difference in Gibbs free energy, delta G value of the G folded and G unfolded, is basically the Gibbs free energy of a folded protein. So the factors which affect the stability of the protein are the relative free energies of the native and unfolded state. So the larger will be the positive delta G, the more stable will be the protein for denaturation. So if the delta G is very high, then the protein will be very stable. It will not denature so easily on high temperatures or harsh conditions. So when we talk about protein stability, how to find out that what is the stability of the protein, and to find out that what will be the stability of the protein, or when we are comparing between two proteins, that what could be the stability of these two different proteins, which one is more stable and which one is less stable. To get all these answers, we need a denaturation curve. For getting the idea about the protein stability, we need to derive a denaturation curve of that protein. And the denaturation curve, if suppose that if we're plotting the graph between a fraction folded or unfolded versus denaturant or temperature, increasing denaturant or temperature on x-axis, then there is a sigmoidal curve which is obtained. This sigmoidal curve has this is a two-state model which shows a monophasic curve, and in this if you see on the fraction unfolded, this is fraction unfolded which is plotted. When the fraction unfolded is somewhat like 0, then what is basically happening that with increase in denaturant or temperature, this fraction unfolded grows and it reaches maximum up to 1. So if you plot the graph, then this graph has three distinct phases. The first phase is the pre-transition region, which shows the dependence of folded fraction YF on denaturant, and then the second is the main transition region, which shows the variation in Y while unfolding process occurs, and then the third is the post-transition region, which shows the dependence of unfolded fraction YU on denaturant. Denaturation curve can be generated by using following agents. There are several agents which can be used to generate the denaturation curve. So temperature is one of the predominantly used one of them. So if you heat the protein will unfold and you can generate a denaturation curve. So I mean, generally these reactions are conducted from between 25 to 100 degrees centigrade. So by measuring any of the parameters which we will discuss further. Then there are certain chorotropic agents. So guanidinium chloride is one of the chorotropic agent, and majority of the proteins which are even very stable, they also gets completely or nearly unfolded at 6 molar concentration of guanidinium chloride. Then urea is another chorotropic agent, and majority of the proteins, they depending on their stability, they unfold between 0 to 8 molar of the urea. However, urea is less potent as compared to guanidinium chloride. Then pH is another agent. Suppose that if we are bringing the pH less than neutrality or from pH 7, or increasing the pH towards alkalinity. So both these are denaturing, and most of the time, majority of the proteins, there are some proteins which are very stable to pH, but there are certain proteins which are very susceptible to pH and they unfolds. So the pH can also be used to generate the denaturation curve, and then the pressure is also used. Sometimes pressure is also used to generate the denaturation curve. So denaturation curve is basically a curve plotted between the fully folded form and fully unfolded states of any given protein. So to plot this denaturation curve, we can use several parameters to analyze. So against denaturants, either we can analyze the some of the parameters. So the first parameter can be the tertiary interactions or tertiary structure. The second parameter can be the loss of secondary structure, and then the third parameter can be the loss of enzymatic activity. So any of these parameters can be used to plot the denaturation curve, and the process of protein folding is reversible. So a renaturation curve can also be generated by measuring same parameters like loss of tertiary interaction or structure, loss of secondary structure, loss of enzymatic activity. Majority of the time for plotting renaturation curve, people use loss of enzymatic activity. So renaturation curve can also be plotted because the process of protein folding is reversible. So we are talking about the first parameter. If suppose that we are measuring the loss of tertiary interactions or structure against denaturant concentration, then what basically happens, like what could be those probes which gives the idea about the loss of tertiary interaction or loss of tertiary structure. So tryptophan is a fluorescent amino acid which is generally buried in the core of the protein. So whenever the denaturant is the concentration of denaturant is increasing, what is basically happening, the tryptophan, the protein unfolds, and the tryptophan, which is buried in the core of the protein molecule, it comes out, and because it comes out, the solvent in its vicinity changes, and with change in its the solvent in its vicinity, the emission maxima of the tryptophan is also changed. So in the given example, you can see if the tryptophan emission maxima is plotted against GDMCl concentration. So most of the time, if the tryptophan is completely buried inside the core, its emission maxima is less than 340 or nearly 340. So if you are continuously increasing the GDMCl concentration, the emission maxima will increase up to 350 nanometer. So this graph is plotted, and one can see like the emission maxima of tryptophan in this case, in the case of FPRA, Mycobacterium FPRA, this molecule is showing the emission maxima of 337, and it goes first down, and then it starts increasing as and when the protein unfolds, and it goes maximum up to 349 or 350. So in this case, when the GDMCl is increased, the tryptophan emission maxima is increased, and by measuring the emission maxima of tryptophan, which gives the idea about the loss of tertiary interaction or tertiary structure, a denaturation curve can be plotted. So the loss of secondary structure can also be monitored as one of the parameter for generating the denaturation curve. So the secondary structures are basically alpha helix and beta sheets, which are predominantly used as a parameter to analyze the denaturation curve. So here in this case, again the example of FPRA, the same example, if suppose that if we are increasing the GDMCl concentration, so what is basically happening, that there is loss of secondary structure, and this loss of secondary structure can be plotted from 100%, it goes up to 20%, because at even very high GDMCl or denaturant concentration also, the complete loss of secondary structure does not occur. So what is basically secondary structure? Here in the left graph you can see that there is a negative ellipticity is plotted. So this negative ellipticity is basically at 0 molar, you can see that there is a very nice graph, and when the denaturant concentration is increasing, you can see the ellipticity at 222 nanometer is continuously decreasing. So this value which is taken at 222 nanometer is basically plotted in the right graph, and then which that shows a denaturant curve, a sigmoidal denaturation curve, and so the secondary structure can be used. Secondary structure or alpha helicity at 222 nanometer can be used to plot the denaturation curve. So the third parameter, which is the enzyme activity, but this parameter cannot be used for those proteins which do not have enzymatic functions. There are certain structural proteins, they do not have enzymatic functions, like the actin, which is a structural protein. So this protein, this parameter can be used for enzymes. So in that case, suppose that with increasing denaturant concentration or increasing temperature or with change in pH, because enzymatic activity is also very much dependent on the pH. So change in pH, the enzymatic activity will vary. So with increase in GDMCl concentration, the enzyme activity can be plotted on the y-axis, the percent of enzymatic activity. And again, in this case, in the case of Mycobacterium apiare, when GDMCl is continuously increasing, the enzyme activity goes down, and it goes down to almost 0% by 1 molar of GDMCl concentrations. However, if you will see in earlier slides, the structural loss occur at relatively higher concentration, like 1.5 molar or 2 molar. So enzyme activity is a relatively susceptible thing as compared to structure. So enzyme activity, loss of enzyme activity always occurs much before than the loss of complete structure of the protein molecules. So that is why this may not exactly correlate with other curves, but it also gives idea about the stability, like how stable the enzyme is as far as its activity is concerned. So denaturation curve can be generated by measuring enzymatic activity also. So the techniques for generating denaturation curve can be many techniques. There are many techniques which can generate the denaturation curve. So so some of them are mainly here, like absorbance is one of the predominant technique, where we measure the absorbance of we know, like protein show absorbance at 280 nanometer. So if the tryptophan and tyrosine in unfolded protein, they will come out, they can show more absorption on after denaturation. So absorption can be one technique for measuring the denaturation curve of the protein. Then similarly, the fluorescence is also one of the important technique, and that is one of the most used technique for studying the denaturation curve. So for measuring the fluorescence, you need a fluorescent probe. So tryptophan is the fluorescent probe which is available in the protein, and tryptophan is generally situated in the core of the protein molecule. So whenever there is denaturation occurs, this tryptophan comes out of the molecule, it comes on the surface. So what basically happens, like you measure the emission maxima of the tryptophan, and by using fluorescence, you can draw a denaturation curve of the protein using fluorescence technique. Similarly, there is another technique, CD spectroscopy, which basically focuses on secondary structure. So mainly used for secondary structure, but it can be used for tertiary structure also in near UV range. But in far UV range, at 222 nanometer and 216 nanometer, basically it measures the alpha helical content and beta sheet content of the protein. So by measuring the loss of alpha helical content or beta structural content, we can measure, we can develop the denaturation curve of the protein molecule. So beside doing CD spectroscopy, DSC is also, differential scanning calorimetry, is also a technique which is used to measure the develop or to produce the denaturation curve of the protein. So then there is another method which is called urea gradient gel. So in urea gradient gel, the denatured protein moves differently and the native protein moves differently. So that also gives a very nice denaturation curve in urea gradient gel. Then beside this, if we add some chromophoric or fluorophoric probes, that can also be used to probe the to probe and generate the denaturation curve. So beside this, the another important technique which can be used for measuring the for generating the denaturation curve is NMR. So in NMR you can measure the conformations, and you can predict whether this conformation is the fully folded form or the denatured form, and you can generate the curve at different denaturant or temperatures. So that will give you the denaturation curve. So thermal denaturation is used to monitor to generate the denaturation curve by monitoring any of the parameters, secondary structure, tertiary, or anything. So here in this case, in this example, we have monitored theta 222. That means the secondary structure to measure generate the denaturation curve against temperature. So what basically happens at high temperature, the disruption of hydrogen bonding and increasing hydrophobicity occurs, and due to which the protein gets denatured against the temperature. So there will be once the protein will be denaturing, there will be loss of alpha helicity and beta strand also take place, because like the secondary structure formation occurs, hydrogen bonding plays an important role in formation of secondary structure. So a denaturation curve will be generated. Thermal denaturation should not be used in case of those proteins which are very hydrophobic, or those proteins which have tendency of precipitation, because at high temperature what will happen, the protein will precipitate and then it will not generate a proper denaturation curve. So the temperature at which 50% of the secondary structural loss or tertiary structural loss occurs, that point is called Tm. So the point at which the temperature at which 50% of the structure or parameter is melted is called the Tm. So in this case, you can see that the protein is not being denatured completely. The curve number 1 is not being denatured completely. However, when cation like calcium ions are added, the protein at low calcium ion is still it was stable, but at high calcium concentration it shows complete cooperativity, and the protein unfolds completely and show a very beautiful the curve number 3, if you can see, it shows a very beautiful denaturation curve at high calcium concentrations. So that means a thermal denaturation can also be influenced by the addition of cations or anions, depending on the nature of protein. So differential scanning calorimetry is also a method which is basically comes under thermal denaturation of the protein. So in this case, what basically DSC measures is the heat required to raise the temperature of the solution of macromolecules relative to the required temperature of buffer alone. And when the this required temperature of buffer alone is subtracted with the protein sample, then it gives a beautiful curve which can be seen in the example. So in this example, we have measured the denaturation curve of two different proteins. One is mesophilic protein, and that is the BS-SHMT, and then the other one is the thermophilic protein, that is BST-SHMT. So the good thing in this, that you can one can see a very nice transitions also, like the denaturation is not a two-state transition. It may be, in case of mesophilic protein, it is basically three-state transition, and in case of thermophilic protein, it is a two-state transition. DSC can also be used to directly measure the enthalpy and melting temperature of a thermally induced transition. So at Tm 50%, at Tm at which the 50% protein is unfolded, the delta G is shown to be 0, and dH is equal to TdS. So the other method of generating denaturation curve is using chemical denaturants. So in chemical denaturation, as we discussed earlier, that we basically use either urea, which can go up to 8 molar maximum, and GDMCl, which can go up to 6 molar maximum. So these two cotropic agents are being used for generating the denaturation curve. So what urea basically does, that it dissolves the hydrophobic core of the protein. So that is why it denatures protein at relatively high concentration, because at low concentration the the hydrophobic core is not accessible to the urea molecule, but when the concentration goes high, somehow it penetrates the hydrophobic core, it dissolves the hydrophobic core, and that is why it is less potent chorotropic agent or denaturant. However, GDMCl first disturbs the ionic interactions, and ionic interactions are basically found on the surface of the protein molecule. So GDMCl basically first disturbs the ionic interaction, and at higher concentrations it penetrates the hydrophobic core, and then it dissolves the hydrophobic core. So these two chemicals, they have different mechanisms of denaturing the protein. So based on that, one can see, like the left graph is the denaturation curve of BS mesophilic and thermophilic protein obtained from urea, and the right graph is the denaturation curve obtained using GDMCl. So one can see, like both the mesophilic and thermophilic, both the proteins, when treated with urea or denatured with urea, it shows only two-state transition or monophasic transition. However, when it is treated with GDMCl, the mesophilic protein shows three-state transition, but the thermophilic protein still shows only two-state transition. So, the denaturation curve can also depend on the denaturant. What kind of denaturant we are using, the denaturation curve can also be dependent on that in the proteins which have multiple domains, not one or two domains. So, and the concentration at which whatever parameter we are measuring, if the loss of structure or loss of that parameter occurs 50%, that concentration is called CM value. So as we discussed earlier, that protein pH can also be used for denaturing the protein, and the denaturation curve can be generated by changing the pH buffer, the pH of buffers. So at high or low pH than neutral pH, some protein gets denatured, but some protein are very stable even at pH 1, like we know pepsin, and there are several enzymes, they work at very low pH, like pH 2, so they don't get denatured at low pH. So at extreme pH, what basically happens, like the net charge on the protein gets altered, because different amino acids have different charge on different pH. So due to this, lot of intramolecular charge repulsion occurs, which basically unfolds the protein. Sometimes what happens, like because of change in the charge of the negatively charged or positively charged protein, the ionic interactions are also disturbed, and because of this disturbed ionic interaction, the protein unfolds. Like in this case, whatever example we have taken, mesophilic and again comparison between mesophilic and thermophilic protein. Mesophilic protein shows loss of both secondary and tertiary interactions measured as compared to the thermophilic protein, because thermophilic protein does not loses any secondary or any significant secondary or tertiary structure at pH as high as 11. So when we are talking about the protein stability, it is always like whenever we compare the stability, we compare the mesophilic and thermophilic proteins. So by comparing them, we can differentiate and find out what are the basic determinants which make the protein more stable, or what are the basic determinants which make the protein less stable. So by comparing this, we can get the idea. So thermophilic proteins which show more stability towards denaturants, they generally have lot of arginine residue instead of lysine residue among the positively charged residues. And then not only the arginine, the increased occurrence of alanine in helices has also been seen in thermophilic proteins. Besides this, thermophilic proteins are also contain a very strong hydrophobic core, which is responsible for its strong structure. So each of these alone makes only a small effect. Like the arginine or the alanine, they make a small effect, but when they come together, several such changes, when they come together, they are enough to make the protein more stable. So in general, it appears that there is no single determinant of increased thermal stability. Each protein is a unique case, So I mean, some protein have stability because of something else like disulfide bonds. Some protein may have more stability because of a strong hydrophobic core. Some proteins may have more stability because of lot of arginine residues in its primary structure. So typically involving variations in hydrophobic interactions. There are, I mean, those hydrophobic residues. So hydrogen bonds, electrostatic interactions, metal ligand binding, and then disulfide bonds. So they all play a role in the stability of the protein. So there is some suggestions that even the packing of the molecule also, which play an important role in the stability of the protein molecule. So there are several factors which affects the stability of the protein molecules. So the first one is we are talking about pH. So protein are more stable in the vicinity of their isoelectric point. So majority of the protein and the isoelectric point of majority of the proteins are close to the neutral. So they are stable in vicinity of their isoelectric point. So in general, electrostatic interactions are believed to contribute to the stability of the native state. So for the maintenance of electrostatic interactions, the neutral pH or a desirable pH is required. Like the desirable pH for some of the protein molecules can be as low as 2, which we talked about pepsin. So those proteins will be more stable even at low pH. So then the ligand binding. In some cases, when ligand binds or substrate binds to the enzyme molecule, the enzyme molecule attains a closed state and its hydrodynamic radii gets reduced, and then the enzyme molecule becomes more resistant or more stable. So ligand binding also affects the stability of the protein. Then the binding of cations, because like majority of the protein molecules, they have lot of negatively charged residues on the surface. So if there will be lot of negatively charged residues on the surface, what will happen that there will be negative charge repulsion, and due to which the protein molecule will be in tense state. So if some cation molecule is added on the protein or in the solution, it will neutralize those negatively charged, and then it will minimize the negative charge repulsion. So in that case, it will facilitate the stability of the protein molecule. Then disulfide bonds are also very crucial. Disulfide bonds, mainly in extracellular proteins, like extracellular environment is very harsh. The temperature may vary, oxidation can occur, then there will be a lot of harsh environment the protein has to face, which goes outside the cell, because the environment inside the cell can be regulated, but outside environment is not very well regulated. So the protein has to have something. So such extracellular proteins evolve disulfide bond for their increased stability. So disulfide bond also provides the stability to the protein molecules. So sometimes when we study the relative stability of some of the proteins, and in case of the protein is very homologous, like in this case which we talked about, the mesophilic and thermophilic protein, they had almost 89 or 90% homology. So when they have such a strong homology, and there is like the difference of 10 degrees centigrade in their TM, then we can do sequence alignment, and when we are doing sequence alignment, we can find out that what are those amino acids which may be responsible for stability or the instability of the mesophilic molecule. So by finding out, and if you get this support from in silico data also, this site-directed mutagenesis support from in silico data also, then if we do site-directed mutagenesis or we mutate those amino acids, we may find a more stable protein by using protein engineering tools or techniques. So beside doing protein engineering and increasing the stability of the protein by doing protein engineering, there are several chemical modifications are also available, which can be done easily in the lab, and the protein can be made more stable. So chemical modification is a vital approach, and it is used in engineering for engineering the proteins and enzymes to suit the research, biological and industrial requirement. Then chemical modification modification of the enzyme is often done with intent to enhance the stability and functionality, to suit the different kind of functionality and to suit the stability in various environments. So beside enhancing the stability and functionality of functionality by using chemical modification is also done to achieve following several objectives, like to identify which part of the molecule is exposed to solvent by probing it, and then to determine which residues are important for an enzymatic activity, then to introduce new groups into protein. That is also can be done by using chemical modifications, and then to cross-link. So many times what happens, like you need to cross-link two molecules, like in case of ELISA, we cross-link the antibody with Haas-Redes peroxidase enzyme molecule. So these cross-linking can also be done by using chemical modifications. So there are certain natural chemical modification occurs in proteins. So what are these natural chemical modifications? Like after protein synthesis, when protein is completely synthesized and comes out of the ribosome, what happens, like certain proteins needs to be post-translationally modified. They can, these post-translational modification can be either phosphorylation, it can be acetylation, it can be ubiquitination, it can be glycosylation, it can be glutathione, anything. So depending on the requirement, these proteins, they do have motif which is recognized by the modifying enzymes, and these post-translational modification can occur in those proteins. So what role basically these post-translational modifications play, like phosphorylation and acetylation on phosphorylation which occurs on serine and threonine and tyrosine residues, and acetylation which occur on lysine residue. Many times this is required for switching on and switching off the enzyme activity, as well as the acetylation at lysine residue is required for chromatin remodeling, which is required for the transcription of the genes as well as the DNA repair. Then the ubiquitination which occurs at lysine residues, that is required for the degradation of the protein. So many a times when the protein is not required, the lysine molecule gets ubiquitinated and the protein is tagged basically to be degraded for using proteasomal degradation machinery. However, if the same lysine molecule is acetylated, then the shelf life of the protein is enhanced because the ubiquitination cannot occur, and then the shelf life of those protein will be enhanced. So these post-translational modification have multiple roles inside the cell, and they are basically covalent or chemical modifications only. So selective modification of protein is a very key step in most biological processes, and majority of the cellular signaling processes or regulatory processes are being carried out in the cell or regulated in the cell using these post-translational modifications. So there are several chemical modification approaches, and after learning from the nature of post-translational modifications, there are several chemical modification approaches has been developed, because these natural proteins sometimes they do not fit to the industrial requirement, to the pharmaceutical requirement, etc. So in that scenario, some chemical modification approaches is required to modify the chemical and make them suitable for industrial application. So there are two different approaches. The one approach which modify specific amino acids, and then it is a specific modification. However, there are several other approaches also which does this protein modification non-specifically, and all these modifications basically occur either on the side chains or on the N-terminal end or C-terminal end of the polypeptide chain. So similarly, as nature uses covalent modification approach in proteins to modulate their function, artificial chemical modification methods were developed to fulfill the industrial requirement, like to the requirement of pharmaceutical industry, the requirement of food processing industry, the requirement of industrial applications. So the chemical modification approaches has been developed to develop more stable proteins or more active proteins. So a large number of site-directed reagents have been developed which reacts mainly with one particular amino acids. So they are very specific. They will be reacting to a specific amino acid based on their nature. At the same point of time, there are several other reagents which reacts very non-specifically with various amino acids. So researchers and scientists, they have used chemical modification approach to achieve many applications in which suits the industrial requirement. So they are pharmaceutical industrial requirements. So these applications are like to increase and decrease susceptibility to proteolysis, because like if they have to be injected in the blood, then they should be relatively more susceptible to they should be more resistant to proteolysis. Then to decrease the immunogenicity, like if we are injecting some protein, foreign protein, then the chances will be there that there will be a lot of immune reactions in our body against those proteins. So by doing some chemical modifications, we can reduce the immunogenicity. Like by doing pegylation, we can reduce the immunogenicity of the protein. Then to preserve and alter electrostatic charges. So that is also one of the important requirement. And then to increase the hydrophobicity of the protein molecule, to increase the UV or visible absorbency. Sometimes the protein molecule which has to act as a biosensor, they have less UV or visible absorbency. So if by introducing some amino acids or by modifying some by doing some chemical modifications, if you can increase the UV or visible absorbency of such proteins, that is going to be useful. So to introduce the fluorescent labels. If suppose that we are going to use some protein as a fluorescence biosensor or probe, we can introduce the fluorescence label by doing some chemical modification. And to attach several different types of carbohydrate moieties, sometimes like biotin is to attach the biotin or other biospecific recognition groups like avidin, we can introduce streptavidin, we can attach antibodies by doing protein conjugation. And then we can introduce several things. The cross-linking is also one of the important thing which can be used by doing protein chemical modifications. So to introduce intramolecular or intermolecular cross-links, both to couple already associated species and to join various proteins in order to combine the properties of two enzymes, let's say, we can cross-link them so that the single molecule will have two different properties. Similarly, we can tag some enzymes with antibody for ELISA kind of assays. So there are many applications which can be achieved by doing chemical modifications of the proteins. So in a specific chemical modification, we modify specific residues. So lysine modification is very common because lysine has a primary amine moiety which can react as nucleophile, and then it can be easily modified. So there are variety of reagents which can be used to attack on this primary amine, and then it can modify that lysine for different purposes. So the purposes can be like probing purposes, can be tagging of the protein, or it can be to enhance the stability of the protein molecule. So similarly, as lysine can be specifically modified, there are several other amino acids like those which have carboxylic group, aspartate, glutamate, and the free carboxylic group is also there on the C-terminal end of the polypeptide chain. They all can be modified chemically using a water-soluble compound which is called EDC. So if this EDC molecule is given and the protein is treated with this EDC molecule, it can modify all these free carboxylic groups. So phenol group is another group which is also available, and it is very reactive. So phenol group which is available on the tyrosine, that also acts as a nucleophile, and it can also be modified using diazonium salts. So there are various diazonium salts which can be used to modify the tyrosine which is freely available in the peptide chain. So thiol group is another group which is very reactive, and it is easily available in many peptide protein molecules, cysteine residues these have free thiol group in the side chains, and which is more nucleophilic than primary amine. So there are several reagents which are available which can easily react in oxidizing environment with these cysteine pre-thiol groups, and then they can form a modification covalent attachment with the thiol group, and they may also lead to different kind of applications. So the free thiol groups can also be exploited for several applications in the protein. So pegylation is also one of the chemical modification which provides significant modification of its to the protein in terms of solubility and bioavailability in the organism. Its long chain prevents the interaction of pharmaceutical protein molecule with the degrading enzymes also. So that is another added advantage to these molecules. Like for example, a site-specific pegylation in the interferon was generated via an attachment of maleimide PEG. So this pegylated product has enhanced the circulating half-life and anti-tumor, antiviral activities in the humans. So the self-life is enhanced because it is not being degraded by proteolytic enzymes. So then pegylation can also be used to reduce the immunogenicity, because if the molecule will be pegylated, the chances that it will be recognized and taken by the immune system will also go down, and then the immunogenicity of such molecule will also be reduced. So pegylation is used in reducing the immunogenicity also in humans. However, there are several limitations of chemical modification of proteins. So protein engineering by chemical modification have these very prominent limitations. Like in protein engineering, you can change any amino acid, whether it is in core or it is on the surface. However, in chemical modification, because you are adding the chemical in the solution, it is accessible only to surface amino acid. So only surface amino acid can get modified. Core amino acid cannot be modified. This is one limitation. The other limitation is like only chemically reactive amino acids can be modified, because they will only participate in the chemical reactions. However, chemically inert amino acids cannot modify. By using protein engineering, we can change the chemically inert amino acids also. And then it cannot be selective. Like if we are adding any specific reagent even for carboxylic group, then it can modify even the glutamate, it can modify the aspartate or anything. So it cannot be selective. So these are some of the limitations which the chemical modification has. Beside this, the protein which are treated with the which are undergone the chemical modification, they have to face several organic, inorganic solvents also, which is a really harsh chemical environment. So this is another limitation of the chemical modification of the proteins. So students, let's now summarize what we have learned in this module. So we learned in this module that protein stability can be classified in three, there can be three different kind of protein stability, like physical protein stability, chemical protein stability, and biological protein stability. So we learned mainly about the physical or thermodynamic stability of the protein in this module. This thermodynamic terms, what does it mean? It is the stability of the protein. Stability of the protein is the difference in Gibbs free energy, delta G, between the folded and unfolded forms or states. So the larger and positive the delta G, the more stable is the protein to denaturation. So the information on the protein stability can be derived from denaturation curve of the protein. So studying denaturation curve is very important. Then the artificial chemical modification of proteins can be done to increase the protein stability, to increase the specificity of the molecule, or to increase the activity of the molecule. And these modifications can only be performed on reactive amino acids like lysine, arginine, aspartate, glutamate, cysteine, or tyrosine, or any reactive protein amino acid molecules. But these are the predominant amino acids which are basically used for chemical modifications. So then, but this chemical modification approach have some limitations as compared to protein engineering. What are those limitations? That I mean, through chemical modification, we can modify only the amino acids which are present on the surface. We cannot touch those amino acids which are present in the core, but in protein engineering we can do that. However, further, we can only use reactive amino acids for protein chemical modification of proteins, which is again a limitation as compared to protein engineering. Thank you.